Hello everyone, my name is Mohammed El-Sherbiny, and this is my novel approach to creating hyper-customizable egg models for embryonic analysis. That was a video of 10-year-old me whistling to encourage my quails to hatch. I believe it's important to understand my perspective and passion behind eggs, as my experience with incubating and maintaining 8 quails sparked not only my interest for birds, but life science. It really was the beginning of my journey, leading to today, where I'll discuss my development of a new type of self-healing covering for eggs, a novel device to window and cover the eggs, and the innovative assays I developed to gauge the effects of modification. So why window an egg? To summarize the novel alterations I'm making to eggs and their implications, I made this diagram. An egg with a successful window above its air cell is covered with an adhesive covering that also acts as a resealable port to the embryo. This allows for the perturbation of the egg's content early in incubation. Specifically, invasive procedures such as the aspiration of albumin, injection of exogenous substances, and the novel use of fiber optic and electronic sensory probes. This can allow for the development of hyper-customizable egg models for embryonic research, while also allowing for the improvement of existing applications such as in vivo sexing, in vivo modeling, and chicken transgenesis. In regards to in vivo sexing applications, my covering improves on existing methods by allowing access to the embryo that won't affect the integrity of the egg's closure. Existing procedures require the penetration of the egg's inner membrane and, as a result, affect hatchability. The novel covering would allow for the extraction of a sample without affecting the resulting hatch rate. With the scaling up of the windowing process, the production of sexable, higher hatch rate eggs would be possible. This is especially significant due to the fact that nearly 7 billion male chicks are culled every year by the egg industry, due to the economic uselessness of male chicks. Aside from the ethical and production implications in the egg industry, the research applications of being able to extract samples interest me, because if gender can be extrapolated from a blood or albumin sample, so can a plethora of other interesting parameters such as the concentration of specific biomarkers. In vivo models are already well established for the modeling of cancer pathology. The chorioallantoic membrane, or CAM, is the avian homologue of the mammalian placenta and is a highly vascularized membrane in developing embryos. The cultivation of different tumor cell lines, as shown in the chart to the left, can then be xenotransplanted into this membrane. The process of tumor development and angiogenesis can then be visualized at the organismal level. Suffice to say, this CAM assay requires access to the embryo, and thus windowing procedures are employed. Current windowing for these types of assays do tend to affect viability and almost never allow xenotransplanted embryos to hatch. With the use of my self-healing window, however, the integrity of the egg's closure would be maintained, increasing viability and hatchability, which is not only important for the CAM assay, but also for observing the effects of cancer treatments on maturing chickens, and not just the embryos. Lastly, egg windowing is needed for the establishment of transgenic chicken lines. The search for new organisms to produce recombinant pharmaceutical proteins at scale has been ongoing due to the high cost and long generational interval of existing transgenic animals, such as cows or sheep. Thus, the use of hens and their eggs as a bioreactor for pharmaceutical proteins is promising due to the cheaper maintenance of chickens and their shorter generations. All this research is only possible, however, using a window into the egg, and current methods affect hatch rate of subsequent chickens. Thus, the use of a novel self-healing window could stand to improve hatch rate, allowing for the safe genetic manipulation of embryos for transgenic protocols. Based on all these improvements to existing applications and their possibility of the development of new ones, it is clear that there's a need for better egg windowing. Thus, the goal of this research was to modify an egg in a way that maintains or improves embryo viability and hatchability, and as a result, expands the data and conclusions that can be extrapolated from in vivo experimentation. Thus, the project became an engineering-oriented one, with the prototype needing to window and cover eggs with a self-healing covering better than any existing device. The result of the design process was this: the first of its kind precision-controlled egg windowing and covering device, divided into 3 checkpoints, magnetically held into place using a ball-bearing carousel. Each part of the machine was controlled using an analog control panel that was 3D printed and fitted to turn and operate each tool. As you can see here, this is the inputting and drilling of the egg. I'm using the Z movement that is contained on the analog control panel. I turn on the drill now. The second checkpoint is the decapping of the egg. And this serves 2 purposes: to remove any debris from the site and also remove the membrane held cap off the egg. As you can see, I'm actually lifting up that cap now with the vacuum pump. And the final step is the application of the self-healing cover, which is preloaded into this device using a hydraulic. It stamps it onto the egg, and the output is a windowed and covered egg, as you can see here. The benefits of the device are very evident when compared to contemporary means of egg windowing. Take, for example, the most recent study of egg windowing from the Brazilian Journal of Poultry Science, which only had an efficiency rate of 30 eggs per hour, a manual decapping of the egg window, limited sizes and shape of the window, and human error due to the drilling tool being a handheld Dremel tool. My device had an efficiency rate greater than 60 eggs per hour, a precision-controlled drilling apparatus, decapping and covering of the egg window, which was a uniform circle, and as my efficiency testing on cohorts of 60 eggs demonstrated, user skill acquisition as the device was used over time. Now that I had a successfully functioning device, I wanted to look deeper into the effects of my modifications had on embryo viability. However, due to local farms stopping their sales of hatching eggs in the winter, I was unable to get my hands on fertile chicken eggs. Thus, analogs to embryo viability such as water loss and cover/shell permeability had to be used in order to gauge effects. I devised a relative water loss study in replicates of 5 of each group in order to measure egg average mass change over time. The controls were normal eggs and uncovered drilled eggs, and the experimental groups were differing ratios of self-healing polymer and regenerated cellulose. The most ideal ratio would then be the group used for the dye ingress test, which I had to design from the ground up as nothing like it had ever been done before. Using a plastic well glued to the blunt pole of different egg groups, 500 microliters of blue dye was seeded and allowed to diffuse over the course of an hour. The resulting albumin of each egg was then isolated, and a subsequent absorbance intensity was then measured, correlating to the amount of dye that was able to pass through each egg's covering or shell. The resulting data from the relative water loss study proved to show a mathematical relationship between cover ratio and total water loss. This was shown using the 4 initial ratios tested, 25, 45, 60, and 80%. Using the quadratic regression of those data points and their corresponding total water loss, I was able to predict that 100% coverage with the self-healing polymer would lose 4.08%. After doing a second relative water loss study to verify that value, I obtained an experimental value of 4.094%, which is very close to my mathematically predicted value. Moreover, Figure 22 compares a positive control and 100% polymer coverage, and the resulting p-value from a t-test is 0.46, which is far above my preset level of significance. This reaffirms the null hypothesis, which I was actually after, the claim that this covering configuration and the ideal normal egg values were not different, at least in terms of statistical significance. Now that I had the most ideal cover ratio, the dye ingress test was done, which yielded surprising results in the fact that not only did my cover prevent less dye ingress, but between 2 different groups of covered eggs, the ones that I punctured and not punctured, there was no statistically significant difference between absorbance intensities. This supports the claim of the self-healing abilities of the covering themselves, which I believe is very powerful. In conclusion, the device was successful in its functionality and ease of use, making the subsequent assays possible, as windowing 120 eggs would have been an impossible feat any other way. Moreover, this study found in a novel way that a covering ratio of 100% silicon polymer was the most ideal, as it had the most ideal water loss and prevented even less dye ingress than a normal group. This suggests that normal water loss and even less bacterial invasion would take place in an actual embryo study, which would increase viability and hatchability. In terms of future prototype modifications, a differing movement system that is automated and employs a laser diode is something that could be implemented, as this would improve the already high efficiency. Moreover, regarding the future bio studies on these eggs, whenever I can get my hand on fertile chicken eggs, probably during the spring or summer, I'll definitely carry out an embryonic study using my proposed covering to confirm that it improves embryo viability and hatchability. Then the real fun can begin with the discovery and testing of new types of probes, assays, and techniques that can be used on the altered eggs in ovo, which is something that I already began looking at, briefly, with the use of these customizable egg caps that actually snap onto the windowed eggs. And as you can see here, I have one that illuminates the egg from the inside, one that takes footage from the outside of the egg, and a probe that actually goes into the egg to measure temperature. These are my references. Thank you.